Welcome, my dear friends. Myself is Professor Dr. Deshpande. Today is the 24th of April 2020, and today our topic of Sharira Kriya Vijnan or Ayurvedic physiology is regarding the membrane physiology. Of course, you must agree that this part is totally devoted or dedicated to the modern physiology. But we have in BMS course and post-graduation course, we have the integrated approach. So this membrane physiology is very important topic. Actually, it should be the cell membrane physiology. Okay. So if you don't mind, I can even right now add that particular word. Cell membrane physiology is the more proper word. Okay. So presented by myself, Professor Dr. Deshpande. I am in Ayurvedic medicine. I am MD and also Ayurvedic physiology. Then these are some of my books: Kriya Sharir Part 1 and Kriya Sharir Part 2. This is also handbook is also available in the market to study the rapid revision of Ayurvedic physiology. Then this is my practical book, which is also available by Shantanu publication. This is very important book for the all the medical fraternity clinical examination. It includes modern and Ayurvedic part. And you can see this is my mail ID. You can contact me for my paid services, that is personal or online health problem advice, then lifestyle counseling based on the Prakriti as well as the palmistry. I also do the palmistry. Personal or online classes of medicine and Ayurveda. Then I request all of you, please subscribe my YouTube channel. Actually, what we are expecting here is LAQ, that is describe the cell membrane physiology. This can be your question in university examination. But more probably the question style will be describe transport across cell membrane. Transport across cell membrane. This is something related with Srotasa concept of Ayurveda. Srotasa concept. Utpatti, Parinamana, Vahana and Utsarjan. These are the micro channels at the cell membrane. Okay. So the contents of this PPT, structure of the cell we will discuss, composition of the cell membrane, functions of the cell membrane, types of the channels. Channel means the doors. At the cell membrane, there are some doors. Basically, there are two types of doors, ligand gated and voltage gated. Then types of the transport are basically two types, active transport and passive transport. Passive transport is again two types, diffusion and osmosis. And active transport is Uniport, Synport and Antiport. Then active transport, some of the examples are sodium potassium pump, calcium channels which are in the neuromuscular junction, hydrogen pump, endocytosis and exocytosis. This will be our topic. So these are the keywords of our today's topic. So actually, you know, the cell is a structural and functional unit of the human body. N number of cells are in the human body, but each cell has a typical structure. First is a cell membrane, which is also called as a plasma membrane. This particular membrane separates extracellular fluid and intracellular fluid. Try to understand. Extracellular outside the cell and intracellular fluid. Then there is a cytoplasm, fluid content inside the cell, and there is a chief head, that is the nucleus part over there. Cell membrane, we are basically today is concerned about the cell membrane. This consists of again proteins, lipids and carbohydrates. But the major content is proteins. That's why we always say that protein is needed for the body for wear and tear, to repair. Protein is always repairing. We are eating the Indian curries, pulses, eggs, non-vegetarian food, soya bean, all gives us the protein. Protein is very much essential for the functioning of cell membrane because 55% is a proteinous part. Then snigdha is also needed, lipid, kafakara dravya. So these are both nutrients dravyas. Huh? This is 40% and carbohydrate is only of 5%. What is cell membrane? This is a fluid mosaic model. Some ingredients are moving inside the fluid. So fluid mosaic model. Proteins are floating. Protein molecules are floating in the lipid layer. Try to understand. Protein molecules are floating in the lipid layer. Okay. This is a central lipid layer. Always there is a central lipid layer, which is a bilayer structure. It is formed by this is formed by the thin film of lipids, and the major lipids. What are the names of the lipids? Phospholipids and cholesterol. Here you can understand the importance of cholesterol. We always criticize the cholesterol, but that should not be there. Cholesterol is needed for the body, and basically cell membrane is having this particular central part of lipid layer which contains cholesterol. Two layers of phospholipids are arranged so that hydrophobic, which is not allowing the water, hydrophobic tail part, and hydrophilic, which allowed the water soluble ingredients like vitamins. Hydrophilic head portion of the outer layer face to the ECF. So that particular head part is facing to the outside and tail part is the inside of the cell. This is the structure of the cell membrane. Okay. Lipid layer allows only fat soluble substances. Lipid will allow only fat soluble substances like oxygen and carbon dioxide, but it will not allow glucose, urea, electrolytes. Protein layers, protein molecules are classified as follows: integral proteins and peripheral proteins. Integral means they bind different molecules together, so network is there, that is integrated proteins, and the peripheral proteins. Next, integral protein is for structural integrating, like a cementing. When we built up the wall, bricks are there, and in between we fill the cement. That cement is nothing but the integral protein. Some protein molecules are channel proteins. Channel means srota, so they allow some elements, useful elements, to come inside. So diffusion of water soluble substances like glucose can enter through this particular channel protein. Do you understand? Carrier or transport protein. So these are the different types of protein. You must remember these proteins: integral protein, channel protein, and carrier protein. Actually, carriers means to carry the person, carry the element. So there are two types of transport: active transport and passive transport. Proteins are helping in this transport mechanism. Okay. Then there are receptor proteins. They will just welcome. They will just welcome to particular hormones or neurotransmitter. So they are receiving the outer elements. Okay, okay, you can come with me. I will carry it to the inside. So these are the receptor proteins. Then carrier proteins acts as a pump. Carrier proteins acts as a pump. And there are some enzyme proteins in the cell which do the metabolic reaction. Try to understand, many of the enzymes like pepsin, trypsin, chymotrypsin, erepsin are proteinous in nature. So we have seen there are 6 types of the proteins in our body or in the cell. Now, carbohydrates of the cell membrane, they also attach to the protein and they form compound, glycoprotein. Glyco means sugar. So this is a compound formed by the glucose and protein together, that is glycoprotein. Carbohydrate molecule form very thin and loose covering over the entire surface of the cell membrane. These carbohydrates, they join this and they form the layer which is called as a glycocalyx. Glycocalyx, thin layer of the carbohydrates. Glycocalyx of 2 neighboring cells help in the fixation of cells, because carbohydrates are madhura, kafakara, shlisha alingane. Kapha is always needed to take 2 molecules together. So that is fixation of cells by glycocalyx. Now, what are the functions of the cell membrane? Number 1, protection. Of course, the wall is there. So wall is for what? It's like a fencing around the cell membrane. So it will protect from the harmful substances like bacteria, viruses, toxins. They cannot enter into the cell. Then selective permeability. Anyone cannot enter from any door. There are the different doors which will allow the different people. Like we arrange the gathering in our college. and we have the main gate, then parallel gates. So VIP people are allowed to enter into the main gate, and students and all other parents are allowed to from the another gates. So the same selective permeability. Lipid will allow lipid layer, lipid part of the layer will allow only fat soluble substances. You understand? Carbon dioxide, oxygen. These are the fat soluble. Then absorption of the nutrients. For example, in the villi of the mucous membrane, you remember primary digestion, stomach, duodenum, jejunum, ileum. Proteins are converted to amino acids, and amino acids are absorbed from the capillaries which are existing in the villi. So that villi also contains the cell membrane. Then excretion in the DCT, distal convoluted tubule, in the kidney structure. We excrete the hydrogen ion. Do you understand? Excretion, that is through the membrane. Excretion of the carbon dioxide from the alveoli. Same, this is the function of cell membrane. Absorption, excretion. Then exchange of gases. Just now I have told you. O2 and CO2 are exchanged in the alveolar cell membrane. Maintenance of size and shape of the cell. Typical shape is there. Biconcave, non-nucleated is a structure of RBC. Do you know? It is non-nucleated, but biconcave. So each and every cell has its typical shape. If that shape is damaged, then that is pathology. Do you remember sickle cell anemia? You know sickle cell anemia? That RBCs are like a shape of the RBCs are like sickle. Koeta Marathi Made. It's like a sickle. And because of that abnormal cell membrane, that RBCs become fragile, easily break, and hemolytic anemia takes place in the sickle cell anemia. Hemolysis. So this is maintenance of size and shape. Next. Now, what is cell membrane is doing physiology? Transport of nutrients and throwing out of waste products. For this transport, there are different channels. So, different doors are there. Okay, and these channels are basically as follows. These are the types of channels. Mainly two types of doors. Two types of doors. DOORS. That is channels. Depending upon the type of the gate, see the word gate. Channels are classified. Number one, ligand gated channels. Ligand gated channels. Ligand is protein. Ligand is protein. So these particular proteins are there on that particular door and they act as a doorkeeper. So whenever proteins are standing on the door, they are called as a ligand gated channels. Okay, but sometimes voltage gated channels, their ions are standing, positive and negative ions, and they charge positive and negative electricity. Current is acting as a doorkeeper. You understand the difference? This is ligand gated channels and this is voltage gated channel. Voltage means electrical current. So charge act as a doorkeeper. Here protein act as a doorkeeper. These two types of channels are very much important. So some channels are guarded by another protein which is called as a ligand. So ligand will be sitting outside the channel and will be keeping channel closed like a doorkeeper. Ligand will be specific for particular hormone or drug medicine or specific neurotransmitter. So this particular protein will allow only specific hormones, only specific medicines, and only specific neurotransmitter. That hormone or neurotransmitter will combine with the protein, that is ligand, and ligand will then open the channel and then particular ion will be moving inside. For example, there are NH channels at neuromuscular junction. Nerve and muscles or synapses are there. There is a neurotransmitter. So that gate is a ligand gated channels. Voltage gated channels. charge, positive or negative charge, will acting like a door, and that is why they are called as a voltage. Voltage is concerned with the current, electricity. Voltage will decide which ion pass through. Now this topic is interesting for the youngsters. Negatively charged will attract positive charged ions. Attraction towards the opposite. If doorkeepers are the male, our friends are standing on the door during the gathering, they will more welcome to their girlfriends. And if girls are standing, they will allow their boyfriends to come in, just by joke. So you must understand this is speciality. Negatively charged will attract positive charged ions. So one of the example is calcium channels are there on the nerve membrane. Calcium channels, positive ions are there. Okay. Now, actually the transport, if it is the SAQ, then you can start directly from here, SAQ. Transport was passive transport and active transport. Passive is also called as a downhill transport. You understand? Downhill. We are climbing to the uphill and we can come down from the uphill. Okay. When we are climbing, it is more require some energy. Climbing hill. But when we coming down, we are running happily. Oh, I can run easily. Okay, do you understand? So passive means coming from up to down, getting down from the hill, getting down from the stairs. Getting down from the stairs, getting down from the hill is easy. Remember, that's why it is called as a passive transport. This will not require much energy, will not, remember, will not take energy. No energy, automatic. Will not need a carrier protein. Our grandfather always take the help of the grandson while climbing up the stairs. Hey, jara mala haath de, please give me a hand. Please give me stick while climbing up. But when he is coming down from the stairs, he will say, nahi, आता माझं मी उतरतो. नाही, आता माझं मी उतरतो. Okay? So this is something like that. That they need a carrier protein support. I'm sorry, they will not need. I should be here. Will not need. When you are coming down from the hill, you will not need any support. Then energy is not required. So ATPase enzyme. This ATPase enzyme is required to break the ATP molecule to generate the energy. But downhill passive transport, we don't need any energy, so there is no need of any enzyme. No need of carrier protein, no need of enzyme. Why? Because passive, we are getting down from the hill, getting down from the stairs. Very easy to understand. Important types of the passive transport: number one, diffusion, and second is osmosis. You must remember these names of the types. So what is diffusion? It is a movement of particles under random thermal motion. It's a movement from here to there. Moving from one cell to another. There is always movement is going on across the cell membrane, across the cell. Okay, so random movement. But there are again three types: simple diffusion, facilitated diffusion, and diffusion through the channel. But you must remember the definition. Diffusion is a movement of particles under random thermal motion. Random thermal at the room temperature. Random thermal motion, movement of the particles here and there. That is diffusion. There are three types: simple, facilitated, and through channels. Now, what is simple diffusion? Movements of the particle from higher concentration to lower concentration. You must remember the definitions. Diffusion, we don't need energy because it automatically happens from higher concentration to lower concentration. For example, oxygen in the atmosphere is more. So oxygen automatically enters into your body. Okay. So oxygen is coming. Now CO2 is more in our body. So it is expelled out automatically. So that is a simple diffusion without any energy, without any enzyme. Okay. So this is called as a O2-CO2 exchange is an example of simple diffusion. I can give one example from our local social event. If you have traveled sometimes to the Pune or Bombay with the local, this is a local train running from the nearby stations. Uh, Bombay people always know there are the locals are going on. Huge crowd. Forget about this corona pandemic, but before normal routine, these trains are full of people, full of people. And there is always one joke. If new visitor is coming to the Bombay and he wants to get down at particular station, for example, Dadar, and he's asking, Sir Malak, please tell me when the Dadar will come. Okay. So before one station, just one of his well-wishers tells, now you go to the door and stand over there. So he said, why? You will automatically get down at the other station when it will come now. You will automatically get down. Why? Because there is a huge crowd in the compartment of the local and that will rush down to the platform. Higher concentration to the lower concentration. Just for the joke purpose, but you must remember the example of atmospheric oxygen pressure is more than the human body. And in the human body, CO2 pressure is more, so it is going out. So this is O2 and CO2 exchange. Now, this diffusion will depend on these things. Number one, concentration gradient. For example, if here is a 10 number, 10 molecules are there and here 5 molecules are there. O2 is 10 molecules, O2 is 5 molecules. So 10 will move to 5. But if there are lot of molecules, 50 molecules, more concentration, they will go fast. Zoom, zoom, zoom. Because concentration is more outside and it is less over here. So it depends on the concentration, quantity. More the quantity outside, it will rush into inside. Okay. Then surface area. For example, in some diseases, our surface of the lung becomes less. And that can reduce the diffusion process. Oxygen will be less. For example, you have the lung cancer. So naturally what will happen? The damaged necrosed part of your lung will reduce the surface area and the patient will suffer from hypoxia. Less oxygen. Why? There is actually atmospheric oxygen is the same. But the entry where it is inside coming, that surface area has been reduced. So naturally diffusion will be reduced. Then it is also proportional to the temperature. In the higher temperature, the movements are very fast. Again, for example, I will give the present example. I am teaching you. There is a pitta, sadhaka pitta, in you and in me also. So I am pouring the knowledge. Some people may feel that lot of knowledge is coming. And it is coming because sadhakagni is there in your brain. Sadhakagni, this is temperature. So unless and until you have the burning desire, do you understand my word? Burning desire is there, you will not understand Dr. Deshpande's lecture. Because that diffusion process will take place only if there is a temperature gradient difference is there. Solubility, how much that particle can mixed with another particle, that called as a solubility. Then this diffusion process is inversely proportional, opposite. If my membranes of the lungs are become thick, then naturally diffusion will be less. No entry, it is resistant, fibrotic changes of there. I had the TB and some part of my lung has been damaged, it's fibrosed. So naturally, I will have sometimes dyspnea because of the thickness of membrane will not allow to diffusion process. Then size of the membrane, size is big, diffusion is easy. Size is less, entry will be difficult. Diffusion will be difficult. So these are the factors which control the diffusion process. Facilitated diffusion. Here diffusion is automatic, but there is some help of the carrier protein. Remember, there is a carrier protein and that will help to entry of that insulin into the cell. So there is one example of facilitated diffusion, that is insulin. So insulin always helps to, that means insulin is entered into the cell because of some protein receptors. Write down the word. protein receptors, they are welcoming. Okay, come insulin, I will take you inside. Of course, insulin will go inside, but there will need some helping friend is there. That helping friend, that is called as a facilitated diffusion. Now you are learning with me. I am teacher is always facilitator. I am helping to understand this knowledge very easily. Actually, it is automatically going inside. But because of the style of the Professor Deshpande, it is facilitated diffusion. Facilitated is mean with the help of somebody. Okay. Next, sometimes diffusion takes place with some particular space area, particular channel, that is called as a diffusion through channels. For example, potassium will be moving from inside to outside. Potassium negative is more. Sodium is outside and potassium is inside. That is called as a resting membrane potential. Cell membrane has positivity and negativity. Positivity outside, negativity inside. So potassium, sodium plus, Na plus outside, K plus is inside. Okay. So if K is more, then it can go automatically outside. But it cannot go anywhere outside. It has some specific channels to go potassium out with the process of diffusion. So that is called as a diffusion through the channels. Okay. Next type of the diffusion is osmosis. I am sorry, passive transport is osmosis. What is osmosis? First we have seen higher concentration to lower concentration, diffusion. Now what is osmosis? Low solute part to the higher solute part. Here you must understand what is low solute and what is high solute. Water and electrolytes, waters and minerals, क्षार, पाणी आणि क्षार यांचं प्रमाण, that is a solvent. So whatever mixed in the water, whatever the elements mixed in the water, that solution becomes the solute. So when kshar, minerals, all these compounds are less in the fluid or in the solvent, that is called as low solute. And when there is a more salts in the water, that is called the higher solute part. Now you can see water absorption through the loop of Henle. This is the kidney structure. There is a loop of Henle. Now urine is formed. Do you understand? Urine is formed. In that filtered plasma, filtered watery part, there are some sodium, potassium, hydrogen solutes are there. But that quantity of solute in the tubule is less than the blood, because outside there is a blood. In the human body, there is a blood. In the blood, there is water and lot of sodium, potassium, hydrogen. And here in the loop of Henle, there is a less solutes. So they are entering from the channel to the blood again. So water reabsorption takes place because of the osmosis in the kidney. So this must understand formation of urine. Now we will talk about active transport. As I have told you, we have to climb the hill, climb up, up, up. So naturally, it's a active process. So it will need some energy. It will need the help of some grandfather, will need the help of grandson, granddaughter, any stick to climb up the hill, climb up the stairs. And as it needs the energy, it will also need the ATPase enzyme. ASE, always remember, ASE means enzyme, which will break the ATP molecules. And what will happen? Because of the breaking of the energy will be liberated. Energy will be liberated. Substances getting pumped against the gradient, as it pushed against the concentration gradient, substance required the energy. So it is like a force. What is pump? It's a force. Try to understand force. So force is needed in active transport. There are three types: uniport, synport, and antiport. Uni means single. Uni means one. Uni means one. Single substance getting pumped. Always remember, active transport pumping. Force is needed. Pumped across the membrane. It is called as a uniport or primary transport. Example: iodine. Iodine is pumped into the thyroid gland. Thyroid gland traps the iodine molecules because of the active transport. Only one substance, only iodine, one substance. Now, Synport. or called as a co-transport. Two substances, Uni 1, Co 2. Two substances are pumped simultaneously. For example, here you can see, I will just correct it. Okay, absorption of glucose and amino acid through the lining membrane of small intestine. I have already talked to you about primary digestion. Duodenum, jejunum, ileum, there are villi, folds of the mucous membrane, there are blood vessels, and proteins are converted to amino acids, carbohydrates are converted to glucose. So these amino acids and glucose both are absorbed in the villi. That is called as a active transport or syn transport, because two substances are pumped at the same time. Next, anti-transport or counter-transport. One is going here, another is coming here. Can you see my fingers? Up, down. So simultaneously, here you can see, carrier is pumping one substance in one direction and other on the opposite direction. Like NAK pump. When resting membrane potential is converted to action potential. Do you understand? Depolarization. So Na is moving in, potassium is moving out. Simultaneous. Na is coming, because Na is always sodium outside, potassium inside. Na is coming in, K is going out. So this movement is called as antiport or counter transport. So these are all pumping movements. That's why they are active transports. Substances transported by active transport, the names. Ionic forms are sodium, potassium, calcium, hydrogen. They are always pumped. And non-ionic are, that's just now we have seen, glucose, amino acids, and urea are transported by active transport. Next, substances transported by active transport, for example, primary active transport of sodium, potassium occur by a common carrier protein who can carry both. That is called as a sodium potassium pump. This pump transports sodium from inside to outside and potassium from outside to inside cell. This pump is responsible for resting membrane potential. I think sometimes I have discussed about depolarization. This is the process coming back to the resting condition. Are you understanding? Sodium is normally out, potassium inside. Depolarization, K going out, Na coming in. But when RMP, resting membrane, what we want again? Potassium inside, sodium outside. So that's why this pump is working. Next, sodium potassium pump. Reduction in the number or concentration of this NAK pump in the myocardium is associated with cardiac failure. We are talking about pathology. When active transport of NAK pump is damaged, what will happen? Cardiac failure will be there. Potassium will not enter into the cardiac muscles. Then next, excessive reabsorption of sodium in the renal tubule. Again, this is a failure of this NAK pump. Excessive sodium is coming inside. Then what is happen? Interstitial fluid, edema will be there. Extracellular fluid will increase, and there will be cardiac load, and that can cause the hypertension. Peripheral blood volume will increase. Do you understand? So these are the abnormalities. We have just talked about the pathology. Now the transport of calcium ions. Calcium is actively transported from inside to outside by calcium pump. This pump is always present in sarcoplasmic reticulum in the muscle. We see the muscle contraction takes place because of this transport of calcium ions. Right now, muscle contraction takes place because of the transport of calcium ions with the calcium pump. Transport of hydrogen ion. Can you see here? This transportation occurs by the carrier protein called as a hydrogen pump. Hydrogen pump is present in the parietal cell of the gastric glands. Hydrogen pump is present in the parietal cells of the gastric glands, and they are involved in the formation of hydrochloric acid. These are some examples of physiological activity of this pump or pathological activity of this pump. So this is the applied part of transport across the cell membrane. First we have seen the theory, first we have seen the introduction, then theory, and then some examples. Okay. Hydrogen pump, another one hydrogen pump exists in the stomach, in the gastric glands for the hydrochloric acid secretion. And you know why hydrochloric acid is needed? To convert pepsinogen enzyme to the pepsin. Okay. Another hydrogen pump is present in, can you see, distal convoluted tubule and collecting tubule in the kidney. And what this pump is doing? They are secreting hydrogen ion from blood into the urine. Remember, hydrogen is a strong acid, and that's why our urine is always acidic. pH is less than 7. Blue litmus paper turns red in urine examination. The root cause is over here. Our body, human body, is throwing hydrogen ion outside of the body through the urine. Through the urine, hydrogen ions are thrown out. Do you understand? So this pump is very useful in the DCT and CT. Okay. Some simple another examples of transport typically seen in the phagocytic cells. Phagocytic, which will kill the bacteria, like WBCs. White blood cells are phagocytic. Phagocytic, remember. Okay. I will just make it a bold. They engulf particle inside. When they are engulfing, taking in, is called as endocytosis. Endo, inside. And giving out the unwanted part is called as a exocytosis. Endo means inside, exo means outside. Okay. So it is ionic concentration difference is very much important to understand the cell membrane physiology. It is one of the property of the cell membrane. Substances more in the extracellular fluid. Which are the substances? This is an oral question, viva question. Which substances are more in ECF, extracellular fluid? They are sodium, chloride, bicarbonate, and calcium. And substances more in the ICF, inside the cell: potassium, magnesium, hydrogen, and phosphates. pH of ICF, intracellular fluid, is relatively acidic. Try to understand. Intracellular fluid is moreover for metabolic process it is required. It is acidic because H plus hydrogen ion are more in the intracellular fluid. My dear friends, I hope you have understood all this cell membrane physiology, and you will definitely answer all the questions related with the cell membrane physiology. So thank you very much for the patience hearing. I will again take you back to the content of the PPTs so that you can understand. We have seen the structure of cell, composition of cell membrane, functions of cell membrane, types of channel, ligand gated and voltage gated, types of the transport, passive transport, active transport. Passive transport examples or types: diffusion and osmosis. Active transport types are Uniport, Synport, and Antiport. Active transport, some applied part: sodium potassium pump, calcium channel pump, hydrogen pump, and endocytosis and exocytosis. My dear friends, you can